Good morning and welcome to Computer Science and Engineering Colloquium Series. I'm delighted to see everybody here at a different time. That's very assuring. Today we have Luke Theogarajan from MIT visiting, our first bionic man here. And Luke has lived in the Northwest before. He was at Intel for a while and worked on the P4, but he has moved a little further away from processor design to a much more interesting topic. So today he's going to be talking to us about bio-ionic neural interfaces. So, Luke.
Thank you, Professor Snyder. Thank you all for coming today. I would like to take you on a small journey today. We will start at the bionic end of things, right there, and move towards the bio-ionic end of things. In more scientific terms, we're going to first talk about electronic interfaces and then move on to ionic neural stimulation. So before we take this journey, I thought I'll give you a map of what we're going to do, how we're going to go through this whole journey. First, we will start at electrical stimulation of neural tissue, and then we'll move and talk about some of the other approaches that people are trying in the field today. I will then talk to you about ionic stimulation, which is something that I've been working on along with electrical stimulation. I'll talk some about the neurophysiology that I've done. I'll talk some about the chemistry work that I've done, not too much because I know none of you are chemists here. I'll touch upon some of the future work that remains, and then I'll conclude my talk with some salient points. So before we sort of begin this journey, we have to ask ourselves the broadest question of all: why are neural interfaces even important? Neural interfaces are the fundamental component that is useful for building devices that can restore function that has been lost due to neural damage. So in sort of picture form, I'm sure you must have either heard or seen the cochlear implant. It is the most successful electronic prosthetic implant out there. It's an FDA-approved device. It's used to restore hearing loss to people who have gone deaf. Recently, in 2007, there was a woman, an ex-Marine, who was fitted with a haptic bionic arm with which she was able to grasp and reach for objects. I've been working on what we call the retinal prosthesis, very similar to a cochlear prosthesis. It's useful for restoring vision to people, to patients who have gone blind due to certain types of diseases. And there's been a lot of interest in deep brain stimulation for helping with Parkinson's disease, especially for tremor control. So before we take this journey, we sort of have to come to some understanding of how neural tissue works, in so much as transistors are the basic building block of circuits. Neurons are the basic building blocks of brain circuits. So I'm going to give you a short overview so that we are all on the same page here. So normally neurons communicate with each other through chemical messages using what are called neurotransmitters. So when a neurotransmitter is released from one nerve to another nerve, there's a binding process that goes on in the receptor, and there's a morphological change, there's a conformational change in the channel, and there are ion flows that happen, and that propagates an action potential. So normally what happens is there is a lot of sodium ions outside of the neural cell and a lot more potassium inside the neural cell. And when you compute the potential, it ends up being a negative with respect to the outside. So when the neurotransmitter binds and the nerve sodium channel opens, sodium rushes in, which actually changes the membrane potential. Now the thing with this particular channel is that it's also voltage gated in itself. So what ends up happening is as the sodium rushes in, the membrane potential changes, which actually makes the channel open more. So it goes into this positive feedback loop that goes in and creates a very sharp spike. Then at some point it closes and the potassium channel will open and the potassium rushes out, which actually goes into a negative feedback loop, which will restore the membrane potential back to baseline. And this happens over and over again. When you see things, when you hear things, when you touch things, this is actually what is going on. So we being engineers, I mean, we sort of have to develop a model. If you have any hope of understanding a system, we need to develop a model. And back in the 50s, Hodgkin and Huxley they came up with this really nice model for a patch of neural membrane. So what they did was they modeled the whole thing using lump circuit parameters. These conductances are actually nonlinear conductances modeled by that equation there. That is the potassium conductance, that's the sodium conductance. This is the capacitance across the membrane. And these small batteries are the resultant. We just saw in the earlier slide how there was an ionic gradient across the cell. Now, if you have a charged species that's going across a semipermeable membrane, then you're going to have a drift diffusion balance, which you can actually come up with the Nernstian balance, which will give rise to a potential. So that's what that models. All right, so now we have a model. Well and good. So now we can start to think about how to interface a nerve to non-biological inputs. We've been thinking about biological inputs so far. Now we're going to take a step further. Now that we have a model, we have a handle, we can think about how to use non-biological inputs. Well, what kind of non-biological input would you choose? Well, with the kind of advances we had in microfabrication and VLSI design, in electronics in general, the best thing to sort of think about from our perspective is to use electrical inputs. So that's what we're going to try and do. So our goal basically is to raise the membrane potential, this VM, from its resting value so that it gets that sodium channel into its positive feedback mode so it makes the cell fire. So basically we want to make that VM larger. So normally what one does is that you apply a stimulus, and we will talk about what that stimulus is in just a second. And if the stimulus is high enough so that it can actually overcome the barrier and set that whole sodium channel into positive feedback, it will actually propagate an action potential, which will then get communicated to your brain and be interpreted as information. So ideally what you would like to do is sort of put a battery across the membrane. I mean, that would be the easiest thing you could do. Unfortunately, you can't do that because all these conductances, even though they're sort of modeled as conductors, they are very specific to the types of ions that they conduct. They're not really electrical conductances. So to an electrical input, they're completely absent. So the only thing that is accessible is this membrane capacitor. The other thing you can try and do is sort of punch through the cell, but that would kill the cell. But if you want to do that in a dish, it's okay. But if you want to do that in a live animal, you can't really do that. So what we're going to try and see is if we can use this capacitor to some advantage and try and interface with the cell. So in practice, what we do is we place two electrodes. One is a stimulating electrode and the other is a return electrode. And you pass current between these two electrodes. So what happens is some fraction of that current is going to go through the nerve membrane, and it is going to charge up those capacitors, which then will change the membrane potential, which will actually get the sodium channel that goes into positive feedback, get the cell to fire, and therefore you're able to electrically couple into the cell. So we've been talking about very broad things here about how the biology of neural cells. We talked about the electrical interface to neural cells. Now we're going to sort of be a little bit more specific and talk about retinal prosthesis. Before we talk about retinal prosthesis, I want to sort of give you a brief, like 2-minute overview on how the retina actually works. So light comes in, hits the rods and cones, which then convert the light information to neural information, such as neurotransmitters. There are levels of neural processing that happen, and eventually it gets to the last layer, which is the ganglion cell layer, and the axons, which is the long part of the neuron, form the optic nerve, which eventually sends information to your brain. So that's actually what goes on in a visual process. So there are some people, some patients who have actually go to blind due to diseases such as age-related macular degeneration and retinitis pigmentosa. And in both these diseases, there's photoreceptor loss. So what actually happens is that there is no way for the light to get transduced to neural information. So this cascade is completely blocked. So this eventually leads to blindness, and they can't see anymore. So we just saw that we could actually take an electrical input and interface it to a nerve. So how would a retinal implant work? Well, we would basically electrically stimulate the remaining healthy layers that are present in the retina using brief biphasic pulses. The pulses are biphasic so that you don't cause any unneeded redox reactions. This will provide what are called phosphines to your brain, and phosphines are what are termed seeing stars. If you ever hit your head really hard, which you should never do, and you see these spots of light and these flashes of light that people call seeing stars, that's exactly similar to a phosphine. We hope that a patient will sort of put these phosphines together and form a useful image. So before I joined the Retinal Implant project at MIT, we were lucky enough to actually have a group of volunteers come and undergo these acute experiments so that we could actually test the theory of whether an electrical implant will be able to restore a useful level of vision to these patients. So the results were very encouraging. I would like to sort of give you a brief overview of what we did. So what happens here is you make a small incision in the white of your eye. You basically cut your muscles out. You make a small incision. You slip an electrode array into your eye. You weight it down with a gold weight so it sits on top of your retina nice and flat. You pass current through the electrode array, and then you give a book to the patient and ask them to draw what they saw so that you have a record of what they perceive. So this is a picture of where the electrode was placed on the retina. That's a fundus photograph. It was a 4x5 electrode array of iridium oxide electrodes. Iridium oxide is a state-of-the-art electrode array material today. And here were the results. So when they gave 4 spots of light, 4 spots of electrical stimulation on the electrode array, the patient was able to see 4 distinct percepts. So the results are very encouraging. It seems that we can restore a useful level of vision to patients who have become blind due to these diseases. So how will a retinal prosthesis sort of look? I mean, conceptually, how will it look? So you have a pair of coils that transmit data wirelessly from a camera, and the image gets transformed to electrical information. It gets transmitted wirelessly to the implant. You stimulate the retina, and eventually the person will see the percept. So that is sort of the cartoon overview of how a retinal prosthesis will work. So surgically, what we decided to do as a group was to sort of move away from sort of putting all the electronics inside the eye. We wanted to put most of the electronics outside on the eyeball because we wanted to maximize the space for things like inductive coils and such. So the way we decided to do this was put all the electronics on the eyeball, make a small incision at the back of your eye. It's called the sclera. You make a small incision. You inject a solution which actually blebs the retina. Retina is held down by a very weak force. So if you put a liquid in there, it will actually lift up the retina. You can slip in your electrode array, let the retina heal, and then you've gotten your implant in there. So when we thought about the problem, and I sort of came up with this sort of architecture for the retinal implant, and we're going to take a look at it more in a detailed fashion in a second. So what we have here is 2 coils. One is a power coil and the other is a data coil. In an implant, you don't want to use batteries, and because you don't want to keep doing surgeries to replace your batteries. So you actually want to be able to power this wirelessly. So we use inductive powering. So we have an outside coil that is the power coil. We have an inner coil that's the data coil. The power coil frequency is at 125 kilohertz, and the data frequency is at a 13.56 megahertz. We talked about the fact that there's a return electrode for the current. That's right there. Now that you have power and data, you need some sort of circuit to process this information and tell the array what kind of currents to put out, what kind of patterns to stimulate. And this is the business end of things. This is the electrode array that gets slipped at the back of the eye. You stimulate all these various points, and that's how you transfer external image information to an internal retinal stimulation. So we looked at the sort of broader overview of how the implant looks. Yes?
What's the scale of the stimulator electrode array size compared to the original rods and cones?
The question was, what is the size of the electrode array with respect to the size of the rods and cones? That's a very good question. And if you'll give me like 5 minutes, I'll answer it in succession. If you want an answer right now, I can give you. Okay, all right. So we'll continue on the same path that we were, which is to basically talk about the architecture of the design. So we have a power transmitter that transmits power field here. You rectify it using 2 diodes. You have a positive supply and a negative supply. You have a data transmitter that puts out data information. We're now going to follow the data stream. We first go to the analog front end, which takes out the carrier and gives you demodulated digital data. You recover the clock and the data from that, and it goes into the control logic, which then tells the current driver what kind of currents, what kind of biphasic pulses to put out. So as part of my PhD, I designed this chip, which is the stimulator chip that is there in the retinal implant today. This is what they're using in the animal implants. It is a 2.2 by 2.2 millimeter chip. It has all the different blocks that I'm going to talk about in a second. You have the analog front end, which has your CMOS detector and a single to differential into converter. So this is going to be a little bit heavy into circuits for the next 20 minutes or so. So I hope those of you who are not like circuit experts will bear with me, and those of who are will get excited and ask me questions. So you have a clock and data recovery block, which is an adaptive bandwidth DLL. DLL is a delay locked loop. It's very similar to a phase lock loop. You have control logic, which I won't talk much about. The programmable current sources I'll touch upon. The electrode bias is actually used to increase the charge capacity of the electrode. So you actually bias it off. If you look at the cyclic voltammetric curve, you can bias an electrode off of the main 0 point to get more charge transfer. I've sort of discussed this work at ISSCC in 2006. I just got my paper accepted to JSCC day before yesterday. So let's begin to the nitty-gritty details here. We'll first start with the Asx detector, which is basically an envelope detector. An Asx is a fancy way of saying amplitude modulation with digital data. So your carrier sort of goes high-low, high-low, high-low. So that's the shift keying aspect of it. What you really want to do is sort of take out the envelope. So if you take a step back and you're like, oh yeah, well, back in my first year of electrical engineering, I've done this with a diode and a capacitor. The only problem is with this sort of circuit technique, you won't be able to follow the low-going waveform because diode detectors are very good peak detectors. They're not very good at following the full waveform. So you need to do either some sort of leak technique to compensate for that, or you need to think of the problem differently. So that's what I did here. So I designed the CMOS circuit that does the envelope detection for you. It looks very similar to a differential pair. The only difference is it has two transistors which are wired OR together. So what does this do? So source coupling in general in any differential pair, source coupling leads to this following of the greater of the two signals, right? So if you have a differential signal coming in, what's going to happen is this source coupled node is going to follow the greater of these two, which is going to give you a full wave rectified waveform, which if you do a negative feedback loop and then filter the carrier, you will end up with your envelope, which is precisely what you want to do. Now, obviously, it's always good to sort of model these things. You can actually model the switching function as a multiplication by a square wave, which is a convolution in frequency space. You follow with a unity feedback loop right there. You can go through all the nitty-gritty math, which I am not going to do here. And voila, you actually do get the envelope information recovered from the waveform. So there are the experimental results from the chip. Here you have the carrier waveform going in, and that is the recovered envelope. All right, so now you've gotten your envelope. You're well and good. Everything is good. But the only problem is it's a very small waveform. So you need to sort of convert it to digital levels. Again, if you think about the problem, you're like, all right, that's easy enough. I'm going to put it to a comparator, compare it to some known reference. And when it goes higher than the reference, it's a 1. When it goes below the reference, it's a 0. The only problem with that sort of approach is that as the reference level moves, the edge of your waveform will widen or shrink. So this will cause pulse width variation. Now, when I talk about how information is encoded, I actually use pulse width as a modality for encoding information. So this would be a disadvantage if I were to go down this path. So the better thing you can do is sort of generate an exact inverse of the waveform and then use basically a comparison and use the zero crossing as your switching point, which will keep your pulse widths intact. So how do you actually do that? So the best thing you can do is use feedback. I'm a very strong believer in feedback. You need some sort of invariant. If you want to have a feedback loop, you need to have an invariant. You need to have some sort of reference you can compare yourself to. The height of the waveform, even though the pulse width varies, the height stays the same. So you can use that as your invariant. So when I thought about this problem, here's what I came up with. And I call this the peak lock loop. And it'll become very obvious why it's called a peak lock loop. So you take the incoming waveform and you buffer it as is. You also take the incoming waveform and invert it with respect to some arbitrary reference. You get that. You compare the heights of these two waveforms, and using that difference, you either charge up or discharge this capacitor, which either shifts up this level shifting node or shifts it down. So it takes the two waveforms which are far away from each other and sort of brings them towards each other. So it brings them towards lock, and you end up with a fully differential waveform. And here is like the gory details of the circuit. You have, so normally when you want to do inverting amplifiers and such, you normally think of using resistive op amps and things like that, but it's better not to do that for power reasons. It's better to do what is called a differential difference amplifier, which uses current subtraction to do its computation. The nice thing about the circuit is that it can do both inversion and non-inversion with the same circuit, so you can use the same topology for two things. You can also level shift everything using the same topology. And the couple peak detector, we talked about the Asx Wave Detector, which is basically uses source coupled maximum following as a mechanism for demodulation. You can use the same mechanism, but without feedback. What will end up happening is it will either shunt the current one way or the other way, depending on which is greater, and that will either charge or discharge this node, which will then level shift this up or down. And that's just basically a two-stage comparator with common mode feedback in between them. So here's a simulation of the locking waveform. You see that the waveforms start away from each other and eventually come to lock, and you get this fully differential signal. My main concern was whether there was jitter on the falling edge to see how much pulse width variation there was, and on two different frequencies there was very little jitter that I could ignore the small variation in the pulse width. All right, we talked about how data was encoded as a duty cycle, as a pulse width. So we're going to talk a little bit about that. So the data is pulse width modulated in the sense that 50-50 duty cycle waveform is a 0, and a 30-70 duty cycle waveform is interpreted as a logic symbol 1. The nice thing about this approach, and it is not my idea, I took it by reading another paper, is the fact that one edge is fixed in time. So the raw data is your clock. You don't have to do any clock recovery. So that actually simplifies your circuit quite a bit. And the data recovery, the way you do that is you take your incoming waveform and feed it through a constant velocity line, and that will do a time-space conversion. And by looking at the different points in space, you can figure out whether there are more ones than zeros and vice versa. If this is not clear, I hope it will become clear in just a second. So you have a constant velocity line that is tuned to one period, which is period t, and you have a register bank that's sort of looking at every point in the delay line. And if you have an incoming waveform, 50-50 duty cycle, it'll come out on the other side after a delay t. Now, the register bank will register that information, and you see that there's a 0-1 transition right about the middle of the delay line. What happens if the waveform is 30-70 instead? Well, it'll do the same thing. It'll come out at a period t later, but now the 0-1 transition happens at a later point in the delay line. And this actually tells you whether it's a 0 or a 1, and that's how you basically decode the information. I told you that this constant velocity line was tuned to exactly one period, and now we're going to look at how you actually tune something to exactly one period. So you use what is called a delay locked loop. A delay locked loop is very similar to a phase lock loop. A phase lock loop locks phases and intrinsically has an integration, because phase is an integration of frequency, and hence is a two-pole system. A delay lock loop only does delay locking, which basically means you take a clock, you delay it, you compare these two, you using a phase detector, and then you charge or discharge this node and use that voltage to control the delay of the delay line, so that both the delay of the line and the incoming are exactly one period, so the edges come into lock. So that's the idea. So here is a problem that we need to address right there, which is, I just told you that you don't want to use batteries. You want to be able to use power coils and things like that. It's a very unregulated power supply. You have a lot of power supply noise. And so you have to be careful how you do this design. So if you go to any analog designer and say, oh, I want something that's power supply independent, they'll say, well, do it differentially. The only problem with doing differential-based design is the fact that it draws constant current. So you really don't want to do that. So you can take one step back and you say, well, why can't I do something with digital stuff? Okay. Let's say you use an inverter as your delay element. The problem is inverters are notoriously bad at power supply rejection. Whatever is at the supply is going to come at your output. But the nice thing is the delay of the inverter is also controlled by its power supply. So if you regulate the power supply using a differential element, and if all your switching elements are purely inverters, then you've gotten the best of two worlds. You've actually regulated the power supply, and you've gotten a low power design. So that's actually what's going on here. The other thing is the entire system is clock frequency independent in the sense that this can operate at any frequency you want. The entire implant doesn't have an internal clock. So in order to do that, you actually, if you want to do a good design, you do what is called adaptive bandwidth. You change the bandwidth of the loop depending on your incoming frequency. So this also incorporates that. Here is the problem, however. So sure enough, you can use a differential element to sort of regulate the power supply. The problem is, however, the differential amplifier itself is a two-stage amplifier, which is a unstable system if you don't compensate it. Now the problem is standard compensation is difficult because if you look at it, it's a nonlinear current draw. So the current switches and then it stops drawing current. So how do you go about compensating something that is not really a truly linear thing? I mean, how do you do something which draws current sometimes it doesn't? Well, one way you can do it is you basically put an extra load that draws constant current, which then burns power. So when I looked at this, I was like, well, there must be a different way to do this whole problem. So I chose to do what is called active compensation, and that's what I'm going to sort of talk about next. So this is a combination of ideas that I've taken from different places. So we have a two-pole system that's unstable. So you have a two-stage amplifier right here. What you would really like to do is sort of put in a 0 in the feed-forward path so that things get compensated. So you cancel one of the poles by putting in a 0, basically. Now, if you look, you can't just go in and inserting resistors wherever you want in a design. So you have to be careful and judicious in how you do it. So one way you can think of it is this. Well, if you took a copy of that voltage, so you got a voltage source, then you took that resistor and put it in series with that copy, and then you took the same current that was flowing in that resistor and made it flow through this resistor, this system is entirely electrically equivalent. If you look at it a bit more closely, you see that this supply and this resistor is basically a non-ideal supply. And it's a copy of this node voltage, which is nothing but a buffer. And then you basically insert the current that's flowing in here. You put it all together, that's what you should get. Here is the actual design. I've actually color-coded things so you can, so this red amplifier, all the transistors are in red, the blue parts are all the amplifier things are in blue, and the green, everything is in green. So here is your first stage amplifier, here is your 0 implementation, and there's your second stage amp. This actually does what is called adaptive biasing. So you actually take the control voltage and generate currents so that you can move the bandwidth of the whole system together. So that's what this circuit does here. It's basically VT dependent current source using a transistor as a resistor right there. So all is well and good. So we've sort of done this design. Now we'll have to sort of think about, I just told you that things operate at different frequencies. So I've compensated the system. How do you know it's compensated through all frequency? You have to make sure that your compensation is also frequency independent. So you can actually sit and develop a full model by using the small signal model of transistors. And if you actually reduce the model, you can actually prove that there is an actual 0 in the feedback path. And if you go through the sort of standard canonical forms, you can show that the operating unity gain bandwidth is this. And if you look at the damping, it depends on all the GMs of various transistors. Now the way I've done the design, all the currents through all these transistors are proportional to the same current, which is the current in the delay line. So you can actually cancel out those terms. And you end up showing that the damping constant is completely independent of everything and is only dependent on physical parameters such as mobility, transistor capacitance, and different ratios of different capacitors. So yes, the damping is completely independent of any operating frequency, so you're compensated throughout your range. So the physical picture of what is actually going on, so you have a system with two poles, and you put in a 0 to make it look like a single pole system. If this pole frequency should shift because of the adaptive bandwidth, the 0 will shift with it. And if it should increase, it will do the same thing. So the system will remain compensated throughout the entire operating range. So here is the results from that section. You can see that a 50-50 duty cycle waveform is interpreted as a logic 0, 30-70 is a logic 1. Here you can see that the clocks are exactly one cycle off because the 30-30 is locked to this 50-50, 30-70 is locked to 50-50. And here it operates in two different frequency rates. Like I said, it's basically frequency independent. The nice thing about this DLL is also that it's linear, so you can actually use it as a frequency detector. And even though the system is made up of nonlinear elements, the result is a linear system. The reason for that is you have a square law current, you have an inverse swing of the same voltage, so you get one-term cancellation. The next part we'll talk about is the control sequence. So the chip comes out of reset, it receives 170 clocks worth of data, and then which basically tells the chip what to do, what kind of current to put out, and things like that. And normally when you want to do pulse widths, you basically use some sort of timer, some sort of clock dividers. The problem, however, is if you want something that's frequency independent, you really don't want to do that. So what I did here was I did the arrival of two commands, the duration between these two, to actually signal the pulse duration. So there are no internal dividers, everything comes from the outside. And here are the experimental results of that section. I'm not going to go through very much except showing you that it actually does work as intended. The last part we'll talk about is the current sources. This is standard supply independent current bias. There are digital programmable resistors to make sure that you take care of process variations. Standard cascode current source, you use the cascode as the switching transistor at the same time. And the currents are pretty well matched. So having designed all this, does it actually work? Well, here is the entire result of the chip driving an electrode array that's immersed in saline, powered wirelessly, driven wirelessly, and that's the top trace is the waveform from the secondary coil of the data. Those are internal register values to make sure that there's data integrity. And here is the waveform across the electrode itself that's immersed in saline. An electrode and an electrolyte can be modeled as a series RC. So you can see there's an IR drop followed by the integration of the current. So that's what that waveform is. All right, so we've done the entire design. It seems to work. So why aren't there more people walking around with retinal implants? Why is there not more people with implants walking around? And this comes back to the question that was asked earlier. What is the electrode size with respect to the actual photoreceptor, etc.? We're going to address that in a second. We've been talking about the processing end of things. We've not been looking at the business end of things, which is the actual electrode itself. So when we did these experiments in human beings, we actually also recorded the current that is required. So at 250 microseconds, you need 1.6 milliamperes of current per electrode. Now let me put this in perspective. If you have a 5-volt supply and you have 1.6 milliamperes, you have 7.5 milliwatts. The entire power budget of my entire chip is 10 milliwatts for the power supply. My chip consumes 1 milliwatt. So you have 9 milliwatts. You have a 7.5 milliwatt electrode. You have 1 electrode. All right, that's bad. Let's say we increase the power budget. Let's go to 100 milliwatts. Well, what have you done? You've gone from 1.5 electrodes to 15 electrodes. All right, little better. So let's say we increase the pulse duration because charge is the most important thing. It's not current. All right, so what happens there? You come to about 200 microamps. So that's a factor of about 8. You've gotten to about 120 electrodes. All right, let's take a step back. The eye has 140 million photoreceptors. If you are trying to restore vision with 120 electrodes, you are tough out of luck. I mean, it's going to be a really hard problem to actually do this. So back to your question, our electrode sizes are 400 microns in diameter. The photoreceptor is about 25 microns in diameter. The reason why you need such high diameters is because you have such high currents. You cannot have a small electrode and pass a lot of current. You'll basically do Faradic reactions. So you should be very careful. So why does this even happen? Well, anybody who's played with electrolytes in salt water and ever shocked yourself in salt water knows that saline is conductive. So most of the current that you're sending is not going to the nerve, it's going around the nerve. So the best thing you can do is to get as close as possible, and even that is pretty difficult because there's glial cell formation. There's all these other issues with this thing. So you need to get a lot closer if you want to make this thing work. That's a very hard problem to begin with. The other problem is the electrotoxicity. I mean, at the end of the day, the electron species is not what gets carried in electrolyte solutions. What actually gets carried in electrolyte solutions are ionic species. So if you want to transfer from an electron to an ion, you have to go through a redox reaction, which means you are going to do electrochemistry. If you want to plate your earrings, this is a great process. If you want to stimulate neural tissue, it's not so good. All right, so what some people decided to do is, these electrical engineers, they really do not know what the hell they're doing. We're going to go back to biology. We're going to look at nature and really try and see if we can take nature and use it to our advantage. So their idea was this. They said, well, if there is a layer of cells that is healthy, why can't we take that healthy layer and convert it to look like photoreceptors? If we could do that, we've gotten what we want. So how would you do that? So what they said was, well, what you can do is sort of use what is called channel rhodopsin, which is a algal protein, which is a cation, light-sensitive cation pump. And if you're able to express it in neural membrane, it will receive light, let sodium pass through, get the cell to depolarize, get it to fire. So that's what they wanted to do. So the way you do that is you take a virus, or in this particular case you take an AAV virus, which is an adenosine-associated virus, because you don't really want to use a virus because it will mutate and cause cancer. And so you can take this helper virus and express this, put in the appropriate promoters and have it expressed in the retina. So when this was tried out, the results were spectacular. So they had the bipolar cell as the adjacent level to the photoreceptor layer express light-sensitive channel proteins. The unfortunate thing was, not only was it there in the first layer, it was there in the second, the third, fourth, fifth. It went all the way to the brain and ended up in 2 layers of the brain. So now you have not only a photosensitive layer in the eye, you have an entire eye that's photosensitive, and you have a brain that's photosensitive. And this may be a good way to get nerves to fire, but the problem is you're going to lose centers around receptive fields, you're going to lose a lot of sensitivity. The other thing is it's a heterologous expression in that you're taking an algal system and expressing it in a mammalian in a framework, so you're actually going to get very lot of loss of sensitivity. So you actually have lights that are equivalent to staring into the sun for like perpetually. So those are things that make it difficult. So people are still trying to solve this problem, but this is one approach. And I thought you should sort of know all the different types of things that are being tried out. Some others decided, well, why can't we do molecular gating itself? Why can't we gate the molecular channel itself using a synthetic molecule? So there are these things called azobenzenes, which is basically this double bond N is called an azo group, and the benzenes are basically those aromatic compounds. And azobenzene basically undergoes what is called cis-trans isomerization. Cis means on the same side, trans means on the opposite side. So when you go from this to this using a UV light, you can actually open and close channels, and therefore you can either let potassium pass through or not and get some level of control to the nerve. The problem, however, is that you need a UV light to cause this isomerization, and that's not a useful thing, because at the end of the day you're going to cleave things. I mean, pretty much if you're getting a bond to rotate at this energy, it's a pretty high amount of energy going in. So some people said, okay, this is not a good idea. Let's go back. Let's just take a step way back to the beginning, which is nerves use neurotransmitter, so why can't we use neurotransmitter? Well, that seems like a reasonably good idea. Seems like the right way to do things. The only problem is this. Let's say you took some neurotransmitter and released it. Where do you get the neurotransmitter from? So one thing you could do is you can have a pouch of neurotransmitter. The problem with that approach is that if you have a pouch of neurotransmitter, if your pouch should break, it will basically kill everything in its site, because it will overexcite every cell and everything will die. So the FDA would not be very happy with that kind of an approach. So the other thing you can do is you can cage the neurotransmitter. You can sort of put a cap on the neurotransmitter and then uncage it using a UV sort of light, and then make it only active when you need to. Again, the problem there is you need a UV light source, and you need these sorts of things, which again make it a difficult problem. So when I've sort of thought about this during my graduate school, I was like, there must be some sort of different way to do this. There must be a different way to think about this problem. What is the fundamental issue? You cannot communicate with the interior of the cell using electrons. However, you can communicate to it with the right ions, because at the end of the day, all these are ion sensitive. So if you were able to do it with the right ions, you should be able to do it. So what ion do you choose? So for that, you have to turn back to the question of what actually the membrane potential is maintained by. And like we saw in the beginning, it's maintained by the different gradients of potassium, sodium, and chlorine. Now at rest, the cell is most permeable to potassium. So the permeability of potassium is much greater than that of sodium and chlorine. So you can pretty much cancel these two terms, which then tells you that the membrane potential is entirely maintained by the gradient of potassium ions. So if you were to be able to control this gradient from the outside, which you can, then you should be able to get the cell to fire. What is the advantage of this approach? The advantage of an ionic stimulation is that potassium is maintained by your body in this background solution. You have to figure out a way to get it from the background solution. So you don't have to make it, you can gather it. So that actually gives you a tremendous advantage over a neurotransmitter based approach. And we're going to see some experimental work. Before that, I just wanted to show you a cartoon of how this would work. You release some potassium chloride outside, it will raise the membrane potential, get your sodium channels to go into positive feedback, get the cell to fire. So when I had this idea as a graduate student, of course, I had to like sort of make sure that you did some experimental work to see if it actually does pan out in theory, I mean in experiment. So what I did is I teamed up with a neurophysiologist, and so I did this experiment where I took a 7-barrel glass pipette, filled each pipette with different solutions of potassium chloride and a control which was sodium chloride, and connected to a pressure ejector and put out small nitrogen pressure pulses which pushed out the solution onto the retina. And you can, that's a picture of the pipette on top of the retina right there, and that's in red light because the rabbit does not respond to red light. And here's a typical recording. So you see there's a pressure pulse that comes out, that's where the pulse of potassium chloride comes out. And that's a recording from the ganglion cell. You can see the bunch of spikes that follow the stimulus. I also did a dose response curve to sort of understand the parameters under which this would work. So here's your background, which is about 5 millimolar, which is what your body maintains. If you raise it to about 10, you get stimulation. If you go beyond 25, you get saturation. So your useful range is between 8 and about 22. So let me put this in sort of electrical perspective. A 10-fold increase in concentration is a 60 millivolts. Okay? So if you want to do something that's a factor of 2 or 3, that you're talking about like 1 kT over Q sort of values. You're not talking very much in terms of energy. So the better way to look at neural stimulus information is to look at what is called per stimulus histogram, which basically says if my stimulus occurred here and if my spikes occurred at a point n, when do they occur between a bracket 0.1 and 0.2, let's say, or different type, and you bin this. This actually gives you some sort of correlation between what you did and what is actually happening. So when you do this for sodium chloride, you get this very sparse spectral density of information, which tells you that it's very uncorrelated background firing. When you do 10, you get a local clustering in the beginning. When you do 15, you get this huge clustering. So that's a huge change of information. And at 30, you get the saturation behavior. I also wanted to sort of compare how light and potassium sort of compare to each other in terms of receptive fields, because that's another problem with electrical stimulation, is the focality of it. So light has about a receptive field about 600 microns, so does potassium. So that was all very encouraging. So the next step then is to ask yourself the question, now that you have a platform to think about the problem, how would you go about building something that can actually make a prosthesis? So here's what I came up with after a lot of imagination. So this is what I call a molecular prosthesis. It has 3 main functions. The first is to sequester, which is basically to take the potassium from the outside, put it inside. The second part is to store that in the system. And the third part is to release it on command. So those are the 3 parts. So I imagine that you could do some sort of synthetic membrane that takes the potassium in, it stores it in a local hydrogel, and then you collapse the hydrogel of some sort. You can do it that way. You can do it with a different type of pumping mechanism. Of course, the better thing to do is to do a hybrid device, which actually couples the electrical processing power of CMOS circuitry with the nice biological compatibility of an ionic interface and put the 2 together. And that's what's shown here. So in my graduate career, I thought, well, I had to concentrate on something that I could sort of carve out. So I decided I would concentrate on the sequestering part and see if it's even possible to build something that could actually recognize potassium ions and bring it across. So when you turn to biology, what does biology do? Well, it basically captures an ion using an ion receptor and then releases it, and it's usually powered using an ATP cycle. That's usually how it works. So could we do something like this? So the first thing you need for this is an ultra-thin membrane. So in organics, you can sort of do PECVD, CVD, ALD, things like that to get nanometer-sized things. But in organics, you need to sort of take a step back. It's better to use the solvent to drive the assembly. So it's called lyotropic self-assembly. Lyotropic meaning solvent-driven self-assembly. You must have all sort of heard somewhere in either introductory biology or come across a paper which talked about the bilipid layer membrane, which has two parts, which is a lipid molecule which has a polar hydrophilic head group and a hydrophobic tail. Now, if you put this in water, because of the entropic requirements of these two things, here you get charge compensation. So this wants to be in water, this doesn't. So there's a self-association of the hydrophobic blocks, and there's a minimization of energy, which actually causes this thing to form the cell membrane or the cell wall. But this is a non-covalent interaction. So from an engineering perspective, it's very non-robust. So you can't really take it out of water. You can't really do an engineering thing with it. So you really can't use this concept. So after I sort of looked at this and sort of looked at the literature, I decided on a different construct which is based on a polymeric membrane. Here you have a hydrophilic polyoxazolene, which basically rivals PEG in its biocompatibility. You have siloxane, which is also very biocompatible. The nicer thing about siloxane is that it has a very low glass transition temperature, so it acts like a very liquid fluid core, so it can sort of mimic the interior part of the cell membrane. And then the third block is all the same oxazolene that was on the other side. But the difference is all the three are tethered together. They're all covalently linked. And furthermore, when you do things synthetically, you can manipulate them even more. So after they assemble, you can fuse them together with a UV curing and things like that. The other problem I had was, well, if I were to put in a receptor that recognizes potassium ions just into the membrane, it will eventually leach away because there's a partitioning between the carrier and the water. So in order to solve this problem, here's what I thought about. So diffusion is basically a molecule on an infinitely long chain, so it can move wherever it wants. So what happens if you shorten the chain? So now you've restricted its motion. You can only go a little bit. But if you put a bunch of them in series, then you can have the molecule sort of exchange from one to the other and do what is called hopping diffusion or hopping transport. So what happens here is if there's an ion sitting on the outside, it'll hop from receptor to receptor and go from one side to the other side. So the problem with all of this was I realized that I needed to do custom synthesis. I can't just sort of go and call up Sigma Aldrich and say, oh yeah, make this for me. It's not going to happen. So I was lucky enough that I was able to find a lab. I taught myself chemistry, and I built this system. And here you have the first thing I had to do was to build a copolymer to which I could attach a receptor. Then I synthesized the receptor, attached it to the copolymer, and then synthesized the hydrophilic block, which I then attached to the other side. Of course, initially when you think about the problem, you sort of think it should work a certain way. The only problem is the same reaction that attaches to this end is the same reaction that attaches to the receptor, so it will block the two sides. So the better thing to do is to sort of put two groups that can initiate polymerization and grow a polymer on both sides. So it's a better way to do things. And so that's what I settled upon. And this is the actual details of the synthesis, which I won't go into. I have published this in a couple of places. If you're interested in talking to me about it, I can talk to you later. The next thing I had to do was, having taken all this construct and made this such a complex system, does it actually self-assemble, or does it actually even retain some of the properties of hydrophilicity and hydrophobicity? So the way you do that is you do what is called Electro Formation, and you basically take a polymer film or a lipid film in chloroform, and you put it on top of ITO glass. ITO glass is basically glass coated with indium tin oxide, which makes it conductive and transparent. And you take two such slides, place it in between a gasket, fill it with a sucrose solution, subject the glass slides to an AC field. The field will find defects in the film and make these bubbles called vesicles. And then you can image these vesicles under the microscope, and then you can see whether your self-assembly was successful or not. And here's the results of that. You can see that you get nice spherical vesicles from these self-assembling polymers. And I did two different receptor densities for other tests, but they both are able to form self-assembled vesicles. Next thing I did was to see if these vesicles would actually, what kind of first order responses they would have to ions. So when you take these vesicles and subject them to potassium chloride, they actually change shape from being spheres to being these long floppy tubes. And the reason why this happens is because you have an object that's a sphere, and if you have lateral pressure in the membrane, that's going to relax a little bit, so it's going to go from a sphere to a prolate object. But the thing is, when you incorporate a positive charge and you leave behind a counter ion, you're going to get electrostatic forces, so that minimization will come into your energy equation. So you're actually going to get curvature minimization. So you're going to go like this, curvature change is going to go into a long tube. But because the lateral pressure is so high, it will be a floppy tube that sort of fuses on itself. That's what you see there. The receptor I used was also sodium sensitive to a little bit, so I wanted to check its response. It forms rigid rods instead, but it forms sparser number of rigid rods. To magnesium chloride, it doesn't do any change at all. I also did a spectroscopy study. I did a study under NMR, and here you can actually show that the hypothesis of packing density is actually what leads to the change. And you can figure that out by the fact that, so NMR works on spins, and spins come from, when you do a spin-based system, if the spins are way too high correlated because of packing, you'll get dephasing of the spin, and you'll actually bury the signal. So that will actually tell you how much packing stress there is, or how closely packed a system is. And by studying it under different ions, you can show that only for potassium you actually see certain peaks in the hydrophobic core, which actually shows you that there's relaxation in the core. So I've talked a lot about only one small part of what eventually needs to be done. There's a huge amount of work that remains to bring this concept to fruition. So the couple of things that I wanted to work on were things that will make better pumps. I need to make a low power releasing mechanism, either using some sort of MEMS concept or using advanced supramolecular concepts. And I need to do more in vitro and in vivo testing. One thing I've been thinking about was how do you take inkjet printing technology and create a platform for puffing out small quantities of chemicals onto the retina so that you can actually study spatial profiles in the retina in vitro. Neural prosthesis are obviously very important technical challenges that we face in our lifetime. Electrical stimulation has been really successful, especially in the cochlear prosthesis, but it doesn't seem to extend to other areas that we would like to attack. It seems to be a very hard problem. I've tried to sort of come up with a different way of doing things, which is using ion gradients. I did some initial work which says that it's a possible way of going down this path, but of course there needs to be a lot of fundamental work and a lot of people coming together to make this a reality. These were all the different collaborators I had, people in the mass eye and ear, doctors and surgeons. My neurophysiologist that I worked with was in the Veterans Administration in Boston. I did some of the fabrication work at Cornell Nanofab, and my chemistry professor was at the University of Alabama in Huntsville. And of course, I could have not done any of this if not for all the people who helped me. And my funding was mainly from the Catalyst Foundation, which funded my entire PhD. And I also got a grant from the Veterans Administration to do some of this work.
You talked a little bit about the spatial resolution of your sensors. What's the temporal resolution? What's your research?
So you can do things at 30 hertz, you can do it at 60. It all depends on how the external transmit. There's no real, because the data rate is so high that you can do a lot of, it all depends on the number of electrodes you actually want to stimulate and so forth.
Is that comparable for the self-assembled?
Yes, yes. I mean, for that you need to make a pumping mechanism that is low power and efficient at low speed. So when you think about anything that involves water and things like that, you have to think about diffusion timescales. But the nice thing about diffusion is as you scale down, it actually works much better. But the thing is, strictly speaking, you should do some sort of active transport. So ideally, you have to use surface tension and things like that to actually drive things. So that would be a better way to go.
You can see on the graphs, what was the time scale between the sodium release and the neural pulse?
It's about 30 milliseconds. The question was, how much duration is there between the sodium release and or the potassium release in my case? You're talking about potassium release, or?
Yeah, I think I meant potassium release.
So potassium release and the response, it's anywhere between 30 to 60, depending on how far you are, where you are, how much flow there is. So there are all sorts of parameters. Yes, the one person in the back.
Working inside the eye is very difficult because you have power constraints and all of these wireless issues. Why not remove the eye and work directly with the optic nerve?
All right, that's an interesting idea. And one thing you can do, so there are a couple of things you can do. So let's just think about it very actively. So one thing you can do is you can cut the optic nerve and put an electrode in the middle and have the nerves grow through the array. And for that you need the right sort of promoters. I think you have to use there's a BCL2 gene that you have to be able to trigger for it to regrow. So people are not able to do that yet. The other thing you can do is you can put a cuff electrode on top of the optic nerve. The problem with that is you're only stimulating the surface of the optic nerve. You're not really getting access to the 1 million fibers. So there are 1 million fibers that are going through your optic nerve. So if you're only attaching it to the surface, you're going to only get an access to very few of them. But there is a group in Belgium that actually does this, and they actually have a patient for the last 10 years with an implant like this one. And they've not had very much success, but they've had the best success so far. Yes, yes, please.
Well, one limiting factor in many neuroprostheses has been encapsulation of electrodes. And I was wondering if you have any insight for your new technology about potential encapsulation issues.
So the nice thing is, so I didn't show a little bit of the work that I also did, which is some of the responses to potassium actually go through glial mechanisms. So in fact, all the glia that usually prevent electrical impulses from getting through actually help this process, because there are a lot of antipode pumps in the glia that actually release neurotransmitter when you increase the potassium concentration. So you can use it as a secondary mechanism to release. So in fact, a lot of the disadvantages become advantages in a ionic sort of approach. And the other thing is, there is no real electronics, and there's no real non-biocompatible systems in here that actually get destroyed due to salt water and things like that, which CMOS really suffers from.
I guess the concern would just be if you've, I don't know if you've done any chronic experiments on animals yet, or if there have been any.
We are actually trying to do some right now. So we normally use Paroline and BCB and things like that, but all of these things eventually have a lifetime issue. So the best, to me, the best way to be is to get outside of the electronics business and sort of go with something that's different, that has none of these issues, because ultimately all these polymeric systems have a finite lifetime. I agree with you that it's a very hard problem that people are still working on. Anything else?
Do you have to deal with, since you're using inductive coupling, do you have to deal with any interference issues, for example?
So the question is, if you have inductive coupling, do you have to deal with any interference issues? Well, because the gap is so small and it's a very localized system and the fields are so strong that the interference issue is not a problem from external sources. But
something like a cell phone, which is
no, yeah, because that's the frequency. So if you look at the self-resonant frequency of our coils, it's in the few megahertz region. Cell phones are 900 megahertz, and the amount of signal they couple in is very little. Anybody else?
Okay, great. Let's thank Luke for a really great job.